Hello, second year. Welcome to the Module 10 up until the Module 12 of our Molecular Biology and Diagnostics. And presenting you here today, I am Ma'am Trina. But before we dive into the discussion of your Module 10, let us just have a quick recap of our Period 1 topics. So, nandito, i-recap lang natin ng mabilisan yung mga topics as well as the discussion that are included on your periodic examination. So, we will begin with your DNA structure. DNA, or what we call the deoxyribonucleic acid, it has three major components. And these three major components comprises your nucleotide. Huwag po natin kalilimutan na kapag sinabing DNA, ito yung polymer of your nucleotide. Kasi basically, nucleotide is a monomer. So when we combine this nucleotide plus nucleotide plus nucleotide plus nucleotide, it will result to a polymer. That is what we call your DNA. So pwede rin namang nucleotide is a polymer of RNA. But basically, DNA po muna. Again, going back to your DNA structure, we have three major components. That is what we call your phosphate group. Also your sugar, pentose sugar to be exact. And lastly, your nitrogenous bases. And in this case, deoxyribose ang pag-uusapan natin kasi DNA ang ating topic for today. So your DNA structure, tingnan po natin on our left side. It is double helical in shape. It is anti-parallel to one another, meaning dalawang template strands yun. Kunwari, if the other way is directing on the right side, the other template is directing on the left side. It is always anti-parallel. And then, what is on the exterior part of your DNA structure is what we call the sugar plus the phosphate group. Okay, sila yung backbone or sila yung pinaka-angkla or sila yung skeletal system, sila yung doon nakakabit. Yung mga tinatawag naman nating nasa loob or interior part of the DNA structure. And paano ba nagkakaroon ng connection or how these two functional groups are being joined together? Yun na yung tinatawag nating phosphodiester bond. So if you will look closely sa chemical structure ng phosphate group and sugar, again, let's say meron kang phosphate group dito. Okay, kunwari maganda yung drawing ko. Tapos meron tayo ditong sugar, pentose sugar. 1, 2, 3, 4, 5. Kunwari na lang maganda yung drawing ko. So 1, 2, 3, 4, tapos sa 5, doon nagkakaroon ng connection. Sa 5 ng pentose sugar, and then sa 3 ng hydroxyl group ng inyong sugar. So yun doon nagkakaroon ng connection at yun yung tinatawag nating phosphodiester bond. So kaya po sila magkakakabit dito. Sa backbone pa lang po yun. So paano naman po nagkakaroon ng connection ang ating mga sugar and then the nitrogenous bases. So, hindi naman po sila nagkakaroon ng connection magically. Meron tayong bonds na tinatawag. And that is what we call your glycosidic bond. And N-glycosidic bond to be exact. Okay, doon nagkakaroon ng connection ng inyong mga sugar as well as the nitrogenous bases. And then for the nitrogenous bases, meron tayong tinatawag na adenine, thymine, cytosine, and guanine for the DNA structure. And then always po silang naka-base pair, parating magkapare si A and T, C and G. Okay? And then for the template strand, tignan natin sa left side, paano ba nagkakaroon ng connection yung mga nasa interior part naman ng inyong DNA structure? Hindi naman sila nagkaroon ng connection magically. Meron silang bond. Ayun yung tinatawag natin na hydrogen bonds. Okay, kasi nga anti-parallel yan kung hahatiin mo, gano'n, no? So, paano nagkaroon ng connection ito, tsaka to? Yun yung tawag nating hydrogen bonds. Yung itong dalawang ito, hydrogen bonds yan, yun yung nagko-connect sa adenine and thymine. Pag tatlong hydrogen bonds, yun naman yung nagko-connect sa cytosine and sa guanine. So, that is basically the DNA structure. Meron pa ba tayong i-add for that one? Wala naman na. So let us proceed with the DNA replication. So according to the central dogma of life or the genetic flow of the information of your DNA, nagdadaan muna yan sa replication. DNA replication is the copying of DNA to another DNA, and it is done in a semi-conservative manner in a way that the daughter cells Okay, meron kang daughter cells or young cells, what we call. One strand is from the mother or the old template. So pag naghati yan, pag gusto na niya mag-replicate, may point of origin siya, at gusto na niya maghiwalay, yung isa, okay, for example, meron kang DNA dito. Ito, magiging panibago siyang DNA, tapos meron siyang young. Ito, saan ba ito nanggaling? Doon, sa old. Tapos itong old na nasa baba, gagawa na naman siya ng panibagong DNA, nandito siya. So, ibig sabihin, it will synthesize a new strand, but the old or the mother strand will be remained. So, pag gumawa na naman ng panibagong DNA, nag-replicate na naman siya, yung tinatawag naman na old, masasama siya dun sa susunod na DNA replication. So, it is in a semi-conservative manner. And lagi po nating tatandaan that replication happens from the 5' to 3'. Again, 5' to 3'. And then for the DNA replication, marami tayong minemorize na sobrang daming enzymes involved. So basically, ito lang siya. Pinahirapan lang tayo ni ma'am, pero ito yung pinaka-shortcut niya. So your DNA replication will begin once your parang proteins kumabit na dun sa DNA, siya yung mag-initiate ng pagbuka ng dalawang template strands. And then, meron na tayong papasok na mga tinatawag nating helicases. Parang siya si Pac-Man. Siya yung pagbubuka at kung gustuhin niya na paghiwalayin yung DNA strands. Pero itong dalawang DNA strands na to, matigas ang kanilang mga ulo. Gusto nila magbalikan. So, ang trabaho ni SSB protein, it will stop the re-annealing of those two template strands. Okay? Yung mga single stranded binding proteins natin, yun yung kanilang trabaho. Siya yung tagapigil para magbalikan sila. And then what else? Meron tayong papasok dyan ng mga DNA ligases. Okay, si leading strand, lagging strand, si DNA ligase, it will join the fragments ng mga Okazaki fragments mo sa lagging strand. Your DNA polymerase 1, it will remove the RNA primer, the RNA primer, kasi may mga kakabit na RNA primer dyan. And then, it will replace DNA primer. DNA. And then your DNA polymerase 3, it extends the chain. And then your DNA primase makes the primer. And then, dito napapasok yung mga tinatawag nating mga DNA gyrase for the prokaryotic cells. Sila yung mga pumipigil doon sa pagkukoil ng supercoiling ng mga cells natin. It helps to unwind the DNA. And for the eukaryotic cells, that meron tayong tinatawag na mga topoisomerases. So, we're done with the DNA replication. Let us now move forward to the DNA transcription. It is the transcribing of your DNA papunta sa mga inyong RNA. And what are the minimum requirement? We have tRNA, rRNA, and then your mRNA. mRNA acts as a template. Your tRNA is the carrier for amino acids, and it is the smallest among all RNA molecules. And then your rRNA, it forms the ribosome. So, ma'am, sana ganun lang po kadali. Okay? And then, it is, the next slide is about the summary of your transcription and translation that will begin with your stage number 1. Stage number 1 is when your mRNA is transcribed from a DNA template. So, after nung DNA, kasunod agad nun mRNA na. Kasunod nun, each amino acid attaches to its proper tRNA with the help of a specific enzyme and then your ATP. So, after nun, dito na papasok si initiation, elongation, and the termination. So, for the stage number 3, which is the initiation of polypeptide synthesis, the mRNA, the first tRNA, and the ribosomal subunits, they will come together. Stage number 4 is that for the elongation, a succession of tRNAs and their amino acids to their polypeptide chain as the mRNA is moved through the ribosome 1 codon at a time. 1 codon at a time lang. Kung matatandaan, meron kang mga UAG, di ba? GAA, for example, tatlo yung mga nitrogenous base. Tapos, ang ibig sabihin nun, codons po yun. So, nire-read niya yung mga codons so that these certain codons can code for a certain amino acid. And then, termination, Termination is when the ribosome recognizes a stop codon. The polypeptide is terminated and then you're ready. So at some point, kailangan din tumigil. Magkakaroon na ng stop codon. Hindi naman po forever babasahin niya mga bases na yan at magko-code na lang siya ng magko-code ng amino acid. At some point, makukumpleto na niya yun at kailangan na niyang pumunta. So that is basically your transcription and your translation. So now, let us now move forward to your mutations. Basically, the mutations, meron kang nagkaroon ng permanent changes in the DNA base pairs. And we have 3 examples or 3 major groups wherein it is categorized. The mutations are being categorized. Number 1, the point mutations. It has your missense mutation, silence mutation, and nonsense mutation. Next is the frame shift mutation. Papasok na dyan si insertion and deletion, and then hindi na natin i-elaborate yung iba. So, balikan lang natin ng mabilis si point mutation. For example, meron ka talagang isang sample outcome of the DNA code. For example, this DNA code codes for the liver cells. Okay, may mga codons ka, codons. At some point, hihinto, UGA na. Tapos na. So, meron ka na buong amino acid. Walang problema, walang nabago. Pag nagkaroon ka ng silent mutation, magkakaroon ng due to the redundancy of the genetic code, no change in amino acid is produced. Wala naman pong pagbabago. However, nagkaroon lang ng redundancy or ng similarity for the coding, okay, of the amino acid. Yun yung sa silent mutation. Next naman sa missense mutation, there will be a change in the codon, definitely. Pag nagkaroon ng change in the codon, anong kasunod nun? Tama po, there will be a change or differentiation in the amino acid kasi nabago yung codon mo. Definitely, kasunod yun, mababago yung amino acid. Missense mutation produces a change in the amino acid sequence in product protein, histidine for arginine. It may change the function of the protein or it may not. So, dito sa missense mutation, natatandaan si sickle cell, mayroong nabagong amino acid. Dito po siya. It will fall under the missense mutation. It may have a very serious effect or it may not have an effect at all, depending. Okay, kung magmamanifest yung symptoms doon sa pasyente. Next is the nonsense mutation. It is a change in the DNA base pair. Nagkaroon ng shortcut. So si stop, dapat nandito po yan sa dulo pa. Okay? Sa kanya, napaaga si stop. Bad news is that the nonsense mutation produces a stop codon within the mRNA transcript, leading to a truncated protein. How short the protein product depends on where the stop codon was produced within the mRNA transcript. So balikan lang natin ang ating point mutation. Sorry, nakalimutan ko na point mutation, it is the most common type of your mutation. Single DNA nucleotide base is replaced by nucleotide. It is what we call base substitution. Yun yung point mutation. Evident naman sa tatlong examples natin. Okay? So your mutation, it has its repercussions and consequences as well. But not all its consequences leads to bad results. Your mutation, it can lead to disease, can lead to silent mutation, and it can lead to evolution. Sabi ko nga sa inyo, matagal na panahon na, puso na yung mutation. It is happening because without this mutation, hindi magkakaroon ng evolution. Not a single organism will evolve to a human being. It will not evolve to a plant or to an animal if it did not went or undergone mutation. Walang evolution without mutation. Okay, walang evolution kung walang, tama. And then there are several possibilities in the consequences mutation. It can, but not necessarily result to a disease. So, ang sabi nga po natin sa mutation, as long as nagre-replicate yung DNA, replicate yan, it can be subjected to changes, magkakaroon ng error at some point dyan sa mga cells na yan. Okay, yung mga kagaya ng mga viruses natin, kaya po sila nag-e-evolve, kaya nagkakaroon ng mga different variants, nagkakaroon ng mutation. Kasi nga, patuloy yung replication. Some forms of mutation result to diseases like cancer, and then when mutation happens in the spacer DNA, usually there are no consequences or silent lang siya. maganda pagka yung mga diseases yung ating minomonitor. Maganda na pag nag-mutate siya, nag-mutate, wala masyadong consequences na mangyayari doon sa pasyente. Pero pagkagaya ng mga COVID, SARS-CoV-2 natin na habang nag-mutate siya, mas tumataas yung transmissibility, mas nakahawa yung mga tao. Yun, hindi po siya magandang epekto ng mutation. But however, meron namang mga magagandang effects si mutation, and then yung tinatawag natin na evolution, the improvement of organism in the function. So dati, let's say, meron tayong isang cell dyan na hindi naman ganito yung kanyang function. So as time goes by, nag-replication ng replicate, naging isa na siyang chicken. Okay, so dati, kunwari, chicken lang siya, as time goes by, naging donkey siya. Parang ganun ba? Merong mutation. So for our previous discussion, we were able to discuss the mutations, the DNA replication, transcription, and translation. So nag-umpisa lahat sa DNA and then natapos siya sa inyong mga amino acids or the polypeptides. But for this slide, we will discuss the DNA repair system kung saan, as we all know, our cells or our body, madami tayong na-encounter na toxins and harmful substances each day. So our body has a natural course of repairing the cells, or what we call the DNA repair. The DNA repair is responsible for minimizing the negative effects that the DNA damage has on the cell. Okay? So madami talagang harmful substances na nade-develop at madami tayong na-encounter na harmful substances that can cause our DNA to be damaged. So, we have these three repair systems, naming your base excision, the nucleotide excision, and then the mismatch repair. So, ano yung mga differentiation on each repair system? Let us discuss each and one of them. So, we will start with your base excision muna. Base excision. Ano ang nangyayari sa base excision? The problem with your base excision is it is spontaneous, okay? Chemical or radiation damage to a single base. So, balikan ko lang. Okay, tama. Okay? So, titingnan natin. Meron tayong, for example, DNA template. Here, nagkaroon ng damage dito. So, for the base excision repair, the bases will be removed from their sugar phosphate backbone. Yun na yung nakita mo doon. Bases may be modified by deamination or alkylation. Damage base is called AP site. Ano ulit ang tawag sa damage base? It is called your AP site or aponuric or apyrimidinic site. And then your DNA glycosylase recognizes the AP site and remove its base. Parang yung inyong mga enzymes, parang pupunta sila doon sa nakita nilang, uy, may damage. Okay, let us fix this damage. And then the AP endonuclease removes the AP site and neighboring nucleotides. Okay. And then the gap is filled by DNA polymerase 1 and then the DNA ligase. And the mistake has been corrected. So, the solution for the base excision, basically, ganito lang siya. Our body has a natural course of repairing that certain damage. But if gagawin siya in vitro, wala naman problema. Pero ito din ang isa pa sa mga solution. Base removal by your N-glycosylase or DNA glycosylase. Basic sugar removal replacement. So, pwede pong gawin nyo ni DNA glycosylase. Pwede din ni N-glycosylase. It is the basic sugar removal replacement. Okay, that is basically what is happening in the base excision. Next, we have your nucleotide excision. So the problem with this certain type of DNA repair system is that it is spontaneous, chemical or radiation damage to DNA segment. So what happens in your nucleotide excision? The two bases adjacent to each other form a dimer. Proteins such as UVRA, UVRB, and UVRC are involved in removing the damaged nucleotides. Okay, kitang-kita. They recognize the damaged bases and they will cleave the DNA and remove that particular DNA strand. Tatanggalin niya po yung strand mismo. Dito, yung mga pairs lang, base pairs lang. Dito, tatanggalin niya yung mismong strand. Yun yung nucleotide excision. And then the gap is filled by DNA polymerase 1 and DNA ligase. It is the same with the base excision. ang ganun yung trabaho ni DNA polymerase 1 and ni DNA ligase. They will fill in the gap once na na-remove na yung damaged pair. Also, for your nucleotide excision, it is spontaneous chemical or radiation damage to DNA segment, and then the solution is to remove approximately 30 na nucleotides oligomer, and it will be replaced. Okay, or it will be filled by your DNA polymerase 1 and DNA ligase. So if may just add, our cells are being damaged each day depending sa mga nai-introduce na toxins or mga chemical substances dyan. Yung mga radiation, okay, they can cause our cells to be damaged, as well as the chemicals, radiation chemicals, ito mga damaging agents yung mga tawag natin sa kanila. Okay, they can alter the bases or they can remove your nucleotide. Next is the mismatch repair. The problem with your mismatch repair is that copying errors, single base or 2 to 5 base unpaired loops. So what happens in your mismatch repair? So the MUTs, which binds to mismatch base pairs. Okay, ito siya. The MUT-L, MUT-S yun, MUT-L is recruited and activates the MUT-H or MUT-H. Activation of your MUT-H cleaves the unmethylated strand in GATC site. The segment from cleavage is removed by exonuclease with assistance of your helicase 2 and SSB proteins. Then, the gap is filled with DNA polymerase 3 and DNA ligase. So, dito, DNA polymerase 1, DNA polymerase 1, DNA polymerase 3 naman po for the mismatch repair. Mismatch repair is very expensive and inefficient. What else? Ano pa bang masasabi natin kay mismatch repair? So, natatandaan si DNA polymerase, yun yung trabaho niya. So, habang nagre-replicate ang ating DNA, merong nagtatrabaho doon, which is the DNA polymerase, to check or to proofread kung mayroong pagkakamali habang nagre-replicate. So, kapag nakalusot doon sa DNA polymerase na yun, si mismatch repair naman, okay, doon na papasok yung mismatch repair pag nakalusot kay DNA polymerase. Okay, for the summary of your DNA repair, this is the proteins. These are the proteins involved for the DNA repair for the prokaryote. So, hindi na natin masyadong intindihin kasi for prokaryote naman na yan. And for the mismatch repair also, nakalimutan ko, the solution for this one is methyl-directed strand cutting or the exonuclease digestion and replacement. So kung makikita nyo dun sa module nyo, meron pong tinatawag doon na methyl-directed mismatch repair system. Basically, yun lang din yung kanyang principle. Mas pinahaba lang. Okay, the methyl-directed mismatch repair system. So after discussing the DNA repair system as well as going back to our period 1 topics, which are about your transcription, translation, and as well as the DNA replication, let us now move forward to our next topics. But first, bago tayo mag-move on sa mga susunod na topic, balikan lang natin yung mga DNA damage na tinatawag natin. Okay? Paano po, ma'am, kapag ka masyado ng madami or masyado ng malakas yung damage sa ating mga DNA or on our cells? What will happen when the DNA damage surpasses a certain threshold? So, ang ating mga cells, meron lang yan certain point kung saan kaya nilang i-carry on or pwedeng kayanin yung certain damage na kung saan mag-work on passive repair system. But if the damage, if the accumulated damage is too much, meron ng mga mangyayaring repercussions such as fates, your apoptosis, yung mga programmed cell death, yun yung apoptosis natin, programmed cell death, ang tawag po doon, and yung mga mutations natin such as cancer. Remember that your cancer, it is when the cell starts undergoing unregulated cell division. It may lead to neoplasia and tumor growth. Kapag sobra na yung pag-cell divide, pag-divide ng cell, and it is unregulated, hindi na siya dun sa tamang bilang niya, it may lead to cancer that may lead to neoplasia and tumor growth. So ito mga recombination repair lang natin yan that are applicable pa din when it comes to the DNA repair system. Pero skip na natin siya. And now, let's move forward to your Module 11, which is about your basic genetics. So ang ating unang topic is about your cell cycle. But first, let's define these terms. We have your diploid or your diploid. It is expressed in 2N. It is the 2 copies of each chromosome. Your karyotype displays all of the chromosomes in a cell. Yun yung tawag dyan. Next, your loci or your locus where the genes resides at the specific locations on a particular chromosome. Your allele, it is the form of gene at a given locus, 2 alleles per locus and 1 per chromosome. Next is the homozygous. 2 alleles are being identical. Kaya, di ba, homo, di ba, parang same. Excuse me. Next, your heterozygous, parang mixture siya, di ba, hetero. It is when the 2 alleles having a different nucleotide sequence which may be caused by mutations. You have your phenotype, the observable traits of the individual produced by the interaction of the genes and the environment. Meron ng factor involved which is the environment. Yun yung difference nila kay genotype. Pag genotype, the genetic composition of the individual. And your heritability, it is the capability to inherit a trait from your parent. Next, the dominant trait. It is the one that manifests itself even when an allele is an heterozygous trait. Kaya nga po siya dominant. Next, your recessive trait. One that manifests only when the gene is in a homozygous state. Next, your codominant traits. Both alleles in heterozygous pair are expressed. Example, yung mga blood group natin. And lastly, for the definition of terms, your sex-linked trait, it is when the mutant allele is located on the X chromosome. So, ang mga chromosome, sex-linked chromosome ng ating mga tao, ay X for the female, and XY for the male. Okay, going back, the disease will generally be expressed in males. So, according to the data, mas mabilis mamatay ang mga lalaki compared sa mga babae dahil sa mga sex-linked trait na ito. Yung mga namamanang sakit na namamanifest ng mga males ang mga symptoms dahil po yan sa mga sex-linked trait. Marami pa rin namang mga factors involved on why males are mas madaling mamatay, mas maagang namamatay. Mas maagang namamatay ang males compared to the females. But one factor that has been mentioned is the sex-linked trait. Females with one mutant allele on the X chromosome are carriers of the disorder, carriers lang, and generally do not express the disease, but the normal allele is present. So, ang rationale on why yung mga babae carriers lang at yung mga lalaki yung nagmamanifest ng mga symptoms or ng disease itself, tignan natin dun sa ating chromosome. So, si X female, si XY si male. In females, the effect of the mutation may be masked by the second healthy copy of the X chromosome. So, dalawa kasi yung X chromosome natin. So, pag may nagkaroon ng failure dito sa isa, meron pa tayong backup na isa pang X chromosome. Whereas for the male, isa lang yung kanilang X chromosome. Magkaroon ng failure man dyan, magmamanifest na sa kanila yung disease. Sa ating mga females, it may be masked by the second healthy copy. Baka may tendency na yung isa healthy, yung isa hindi. May backup X chromosome. Sa mga males, wala na po sila. So again, your genetics, it is the DNA within the human nucleus. It is organized into your chromosomes. Sa genetics, pag-aaralan natin ng inyong DNA pa rin naman. It is where the DNA within the human nucleus is organized into chromosomes. Your diploid with 46 chromosomes. Your 22 pairs of autosomes and 1 pair for the sex chromosomes. Okay? And we have 3 billion base pairs. Pakitandaan, we have 3 billion base pairs. And for the chromatin, it is being comprised of a telomere with repetitive DNA and centromere with highly repetitive sequences. So, ang sabi dito pala, balikan lang natin si chromosomes. We have 46 chromosomes, yung 23 doon galing sa inyong mother. And then yung 23 doon, galing sa inyong mga father. So basically, yung 50% ng DNA nyo galing sa mama nyo, and then yung 50% galing sa father nyo. So maswerte na lang kung yung napunta sa yung DNA and the pair of chromosomes ay yung magandang cell or magandang chromosome ng inyong parents. So for the sex determination, people, lahat po tayo, meron tayong 46 chromosomes and it has 23 pairs. 23 pairs are homologous. They are being called autosomes determined by the body traits. One pair is the sex chromosomes. It will determine if yung sex mo, if you are a female or you're a male. And the female sex chromosomes are homologous. It means look alike, labeled as X and X. Pag male naman, sex chromosomes are different, X and Y. X and X sa female, X and Y sa male. So lahat yan, homologous sila, magkakapareho ang kanilang mga pairings. Tapos ito, meron tayong ika-23rd na pair dito, which is for the assignment or the determination of your sex. So now let us proceed with the cell cycle. Mabilis lang ang ating cell cycle. Hindi na ako magdodrawing ng chromosome masyado dyan. And then mag-uumpisa po tayo sa ating G1. G1 phase. The G1 or the gap 1. Dito, anong mga nangyayari sa gap 1? So, unang-una dyan, this phase is where your organelles are being made. Okay? Yung isang organelles, gagawa na yan ng maraming organelles. Kasi nga, ang goal is mag-divide siya into 2 doon sa dulo. So, para ma-divide siya into 2 at kung anong meron yung isang cell, dapat meron din yung isang cell para same sila na merong laman. Kunwari, may ribosome sa isa. Yung isa, may ribosome din. So, para magkaroon sila ng equal amount nung kanilang mga component. Okay, itong cell na to, itong cell na to, may nucleus, may ribosome. Dapat sa G1 phase pa lang gumagawa na ng mga organelles para makapag-ready doon sa cell division. What else? Dito sa gap 1 phase, gumagawa na or nagsisynthesize na ng mga proteins and enzymes that are needed for the DNA replication. Okay, growth and synthesis of components required for the DNA replication, and then gumagawa na ng mga organelles. 1 and 2. Okay, and then also for your gap 1 phase, it is where the repair of thymidine dimers, if ever na may mga errors, na-check pa rin niya yan. The repair of the thymidine dimer. So, tatlo yung functions niya. Number 1, it makes more organelles in preparation for the cell division. Gagawa na yan ng mga organelles. Next, the synthesis of the proteins and enzymes needed sa ating DNA replication. And then, the repair of thymidine dimers. Okay, let us proceed with the next step, paikot. So, bago tayo makarating sa S phase, nakasunod, meron muna dyan G1 or S. G1 or S na checkpoint. So, alam niyo na kung para saan ang checkpoint. So, now, let us proceed with your S phase or your synthetic phase. Nandito na po yung mga ating DNA replication or the DNA synthesis. Huwag kalilimutan. And then, yung inyong diploid magiging, ano na siya, apat na. Magiging 92 chromosomes na siya dito. Ma'am, bakit po papadamihin? Kasi nga in preparation nga po sa cell division para mahati siya ulit into 2. Okay, so yung naging diploid mo magiging ano na siya, 92 pagdating dito. Okay, so next step naman natin is yung G2. Nasa G2 na po tayo, gap 2 phase. Ang nangyayari na sa ating gap 2, meron ng growth. Okay, magre-regulate na yung cell growth. Dadami na yung cytoplasm, the jelly-like structure inside the cell. Diba? Dadami na yon. Para pagka hiniwa mo siya or pinaghiwalay or dinivide mo yung iyong cell, equal yung amount ng laman ng kabilang cell, equal doon sa kabilang side. Okay, dapat kung ilan yung laman ng kabila, yun din yung laman ng kabila. Okay, and then yung tatlong phase na nabanggit ko from the gap 1S and the G2, yun yung tinatawag natin na interphase. Ang gulo-gulo na ma'am, ng sulat mo, interphase pa lang po yun. So papasok na tayo dun sa susunod nating step. So bago tayo pumasok, meron tayong G2 checkpoint pa rin naman. So you know the drill. Next, meron na tayong mitotic phase, the PMAT. PMAT. It will start from your prophase. Anong nangyayari sa prophase? It is where your nuclear envelope are being dissolved. Number 1. Number 2, the centrioles. Yung mga centrioles nyo, it will begin to move. And number 3, magiging condensed na siya. Condensed ba? Parang mas packed na yung mga cells, yung laman ng cells. Next, for the metaphase, PMAT, it is the most distinctively visualized stage. Kitang-kita na talaga na gusto na talaga siyang maghiwalay at ready na for the cell division. Nandito na yung tinatawag nating sister chromatids. Sister chromatids. It is your sister chromatids line up along the equatorial plane of the cell. Hindi ko siya marunong i-drawing, pero let's say meron kang chromosome, yung equatorial, yung parang ganyan, magkakaroon na ng hati. Basta, sa sister chromatids, it will line up along the equatorial plane of the cell. Saan nangyayari yun? Sa metaphase. Next stage is your anaphase. Yun na yung pinaka-action stage of the mitosis. And then magse-separate na yung dalawa mong sister chromatids. Again, sa metaphase, nag-line up yung sister chromatids. Sa anaphase, maghihiwalay na yung sister chromatids. Next po, PMA. Uy, nasa T na tayo, telophase. So, kung condensed pa po ang inyong mga cells doon sa prophase, dito sa ating telophase, sa dulong-dulo na, madi-decondense na, malulusen up na yung cells. Ready na po talaga for the cell division. Na-loosen up na with equal amounts yan, ha? So, lastly is the cytokinesis. So, done with interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. It is the final step in the cell division. It requires pinching of the plasma membrane and the formation of two separate membrane bond daughter cells. Okay? So again, ang ating cell cycle, G1, S phase, G2, tapos interphase yung mga yon, prophase, metaphase, anaphase, telophase. So that is your cell cycle. So ito lang siya, basically, ang mga information that I included